Hi, and welcome to this environmental adaptation of animals. And these few videos that I'm going to show are on molecules and cells in the animal physiology. So it's something that you should already know from the cell biology, from the molecular biology, from the biochemistry, and from the animal physiology. But it's kind of a review for the course. And we will start with the cell membranes. And why we are considering on cell membrane is that not all the predators are running after you. They can just wait for the prey, and they had a decent poison that is affecting very fast, so they don't have any need for running after you or run for their life. So these poisons are usually affecting on the membrane proteins, and that's why we should understand very carefully that how these membranes are working. Okay, if we have a cell, it's surrounded by hydrophobic cell membrane, like in this balloon. And this hydrophobic cell membrane is limiting the diffusion of water-soluble particles, like in this balloon there was water-soluble blue color substance. And when we have a hole in the membrane, the diffusion of this blue stuff is increased through this membrane. And that affects then that, if we have uncontrolled holes in our cell membrane, our cells are dead. So why the membrane is important? Well, everything that we want to have immediate response, fast response, it's affected by electricity. And if we have electricity on the cells, it's controlled by membrane proteins and membrane itself. And furthermore, the separation of the intracellular fluid from the extracellular plasma is essential for the cellular homeostasis. So if we have a hole in the membrane, we have a dead cell. And also, the majority of our predatory-based poisons are affecting by these cell membrane proteins. And that's a very good reason for studying it. And in the membrane, we have membrane proteins. They can be inside the membrane and called integral proteins, and they are embedded in this phospholipid layer. They can go through this membrane completely, as like over here we have an ion channel. Then we have peripheral proteins that are not inside the membrane, but they are bonded to these integral proteins or these lipids. And then on the extracellular side, we have carbohydrate chains that are bonded on the membrane proteins or the lipids. And how this membrane is regulated, or what is the structure of it? Well, first of all, we have phospholipids. In phospholipids, one head is polar and one head is non-polar. We have polar head with differently positive charged group, and then there's a phosphate group with a negative charge. So that's why the polar head can be neutral, or it can also have a charge there, depending on is this polar head having more positive charge or less positive charge than this phosphate group is negative. And then in the other side, we have these hydrocarbon chains that are derived from fatty acids, and they can have double bonds. And if they have double bonds, it means that, it's bending permanently, and it takes a little bit more space, this phospholipid. And, this is the quite often seen structure that, this is kind of phospholipid we have, and then they are making a bilayer together, so this polar head is toward the water phase. This is something that happens spontaneously because we have a long nonpolar area that is always trying to get out from the or as far from the water molecules as possible. And what is the structure of phospholipid? So if in very cold temperatures, it's in pseudocrystal structure. And in these, the carbon chains are all in trans-oriented. So if we are looking toward from one carbon to next carbon axis, so the previous carbon that is toward us is on upward. Over there is the R. And then the next carbon, after that one particle bond, is on downward. So that's why it's making a zigzag in all these pictures. And this will put all the carbon atoms as far from each other as possible. But as you can see in this picture made when it's been detected, the pseudocrystal structure, it's not always zigzag. It's bending, etc. And it can, if it bends, it will turn the carbon chain by 120 degrees. And this can cause that, okay, the phospholipid chains could clash with each other, and that's always something that we, all the molecules try to avoid. But anyway, it can happen that they are bending. And especially when it's a little bit warmer temperature, we have a chill structure. So these carbon chains can turn easier, especially if the following carbon is also turned. That will mean that, okay, one carbon bond will turn to leftward, and then the next one is turned on rightward. So it means that, okay, the chain is continuing in the original direction. And as you can see, also the hydrophobic head group is now turned in warmer temperatures. And then, this is still something that this is not very physiological structure, this chill structure, but in the functional membranes, it's actually liquid crystal structure that is quite far near. And you can see that it's a mess. So it's nothing in the same way as in the school books. No. And actually every phospholipid class has a typical transition temperature from the chill to liquid crystal. So that's why they can be chill or liquid crystal depending on the temperature. And quite often our membranes try to be in this liquid crystal. So in that case, there is not much limiting for the membrane proteins to have different conformations. And there are actually 4 ways to keep the cold membrane fluid. First of all, we can change the membrane phospholipid type. So if we have, for example, 3 different types of phospholipid, we can increase one type in the cold climate and make the fluidy a little bit fluidy more. And then, of course, what we all know, that actually if we are increasing the unsaturation, it will get more fluidy, and there are more double bonds, and it can be fluid also in cold temperatures. Then we can affect on the amount of cholesterol. So a little bit less cholesterol, we can get it more fluidy, but then actually also in high temperatures we need cholesterol for this to support this membrane. So that's why in mammals we have a quite high cholesterol content in some membranes. And then the 4th way, that is a little bit more difficult, is that actually our membrane are not homogeneous. We have different areas. We have these rafts which has typical phospholipids, and then non-rafts where some other phospholipids exist. And we can then change the phospholipids in the raft area or in the non-raft area. And that makes it a little bit messy. But on this saturation and the number of double bonds is the most studied area. And this is something that you should already know, that okay, the membrane saturation depends on the adapting temperatures. So that animals that are living in cold environment, they have more unsaturated phospholipids. And this is an example of comparative method used to demonstrate the adaptation. So just collect different animals and then check that, okay, where they live and how much unsaturated phospholipids they have. And as you can see, in Antarctic species, they have, in phosphatidylethanolamines, they have maybe 80% of these phospholipids are unsaturated, where in tropical species about 60% or 55% is unsaturated. But as you can see on this, there is actually two lines. There are phosphatidylethanolamines and phosphatidylcholines. And this means that if you are studying this, you must first isolate different phospholipids from each other and then check them one by one, that okay, how many double bonds there exist, etc. And the interesting thing is actually that when we are testing the fluidity of this membrane in Antarctic species or the tropical species, when we are having in the habitat temperature testing it, it's quite similar. So it's as fluid as, for example, the normal oil that we are using in the kitchen. And furthermore, it's not only that, okay, those animals that have been living for ages in cold temperatures, but we can actually change the saturation of these phospholipids in our annual cycle. So we have annual changes that can be done in the acclimatization. So these unsaturated phospholipids can be less common in summer, and then unsaturated are more common in the cold season. So okay, here you can see phosphatidylethanolamine. The first one is having no double bond, so that's why it's 18 times 0. And then there's two other ones are the in the middle one there's 22 times 6. It's a little bit longer phospholipid and having a double bond, and then 18 times 1, that is the similar size as the first one, but it has one double bond. And you can see that one is going up in the summer, one is going down. So these animals can change seasonally the membrane composition. And actually they are the same Crucian carps that I've been showing you on previous slides, that actually these one we were able to test it also in laboratory conditions by acclimating the animals in different temperatures. So it's temperature dependent, not dependent on the amount of light available, for example. Okay, but beside these membrane phospholipids, we also have membrane proteins that are very important to understand for several physiological functions. We have, first of all, we have ion channels that are my favorite. We have something that is permitting diffusion of one or perhaps sometimes in completely non-selective, so it can be several different types of solutes that are able to pass the channel through this membrane. And then we have transporters that are a little bit slower ones, and they are bending the thing that okay they want to transport, and then they are changing the conformation, so and then unbinding the protein, so or whatever they are delivering, and then that means that okay they are changing the conformation quite often, and that's why they are much slower. Then we have enzymes that are catalyzing some chemical reactions. We have receptors, and so something to detect what kind of chemicals, for example, we have in the extracellular fluid. And then we have structural proteins to keep the cell structure quite much the same day after day. Okay, these membrane proteins, they can be described in several ways. This is a very common in the science. So these kind of hypothetical secondary structures where these barrels, they are all presenting alpha helix going through this membrane. So these membrane transmembrane areas. And for example, over here we have a sodium channel that has domains. So four different domains with which each have 6 alpha helix. And that's why in these kind of pictures it's very easy to draw that, okay, somewhere over there we have somewhere there we have single amino acid that could be changed in some diseases, etc. And so it's getting it very, we can actually have a very detailed structures described in these kind of pictures. But then we have different other ways, because when we have these kind of 4x6 transmembrane helix, they are always, of course, making some kind of three-dimensional structure. So over here we have the tertiary structure, and in this is a little more messy to describe it, okay, how it's working. And then it can be stylized or make even a tube there going through the membrane, and then add some other areas describing that, okay, what kind of beta subunit or gamma subunit we have there. And so the same thing can be described in several different ways. Or how I like to bake it, I just have these kind of barrels. Over here we have the same sodium channel. So it's just some kind. I will just draw that, okay, there is a arrow indicating that, okay, the ions are flowing, and I will quite often I write that, okay, what kind of ion we have. So it's a sodium ions. Okay, it means that there is a sodium ion channel, and then we have 2 transporters. On the right hand side we have sodium calcium exchanger, that is moving sodium out from the cell, calcium inside the cell, and it can actually work on both directions. So it depends on how much sodium there is inside and how much calcium there is outside, etc. Or then we have a sodium potassium ATPase, meaning sodium pump, that is pumping sodium out from the cell and at the same time pumping potassium inside the cell and using ATP. So we can have different kind of ways to describe it, and quite often in electrophysiology we are just having these arrows to indicate that, okay, now it's this kind of ion channel, etc. Thank you.